Hello everyone, thanks for coming. I would like to start with one question. Do you know somebody suffering from cancer? Personally I do, and moreover I work for a biotechnology company, and sometimes I hear some stories about good people with cancer, and these stories really annoy me. So being a computer scientist or a bioinformatician or a data scientist, how can we help to solve the cancer problem? How can we contribute to this fight? In order to understand this, today we will try to answer the question: how to cure a lot of people if you have only a supercomputer? I will continue after a short introduction of my company. I work for a biotechnology company called BioCAD. We discover, develop, produce, and sell drugs. Nowadays we work in seven countries, especially in the field of oncology. We have 46 products in portfolio and about 16 pipeline. There is a computational biology department in BioCAD where we try to hack life to provide high quality medication for patients. Okay, to answer our question, let's first dive into some history. How drugs were discovered in the past. Often it was made just by trials and errors, or even accidentally. For example, first antibiotic penicillin was discovered completely accidentally by Alexander Fleming, and it was one of the greatest discovery in medicine of all time. As a result, drugs discovered such way had a lot of side effects, and no one actually knew how they worked. Fortunately, nowadays the industry is much more mature, and modern processes of drug discovery can be called rational. After each iteration of drug development, we certainly know which properties are changed and how it results to final efficiency of a drug. Which molecules types will I talk about? My company mostly works on drugs based on two molecules types, small molecules and antibodies. The former are typical drugs that we all get used to. For example, aspirin is a molecule of comparable size. The latter are antibodies, much bigger protein molecules that use natural cell processes as a treating mechanism. Let's briefly look how they work. Let's imagine we have the tumor cell and the immune cell. Tumor cell has its own specific receptors, and there are antibodies, very special immune system agents that bind to tumor cell receptors. Also, immune cell has its own receptors that bind with the other side of the antibody. When this happens, so-called effector function of the antibody is induced. Effector function in our case is to kill tumor cell. So immune cells are able to do this. The most important step of this whole process is binding of the antibodies with tumor cell receptors. We can design very specific antibodies that bind with particular targets. In order to do this, we apply very complex iterative pipeline that could be briefly described this way. We have big library of antibodies obtained from a lot of people and from some llamas. Then we iteratively optimize, we screen our library to get some initial approximations, and then we optimize these molecules with different techniques and laboratory tests. And one of the most interesting stages of this pipeline is in silico optimization of molecules. As I said, we have initial approximations, antibodies. We have structure of our target or antigen. For modeling purposes, we take a small part of the whole antibody. We take the most variable part of it that is responsible for binding with the antigen, and it's enough for modeling purposes. So the aim of the optimization is obviously to optimize the properties of molecules, to increase affinity, stability, to reduce side effects, immune response to the antibody itself, and so on. So both molecules are protein molecules with corresponding amino acid sequences. The optimization itself is done by replacement of particular amino acids with another ones, and after we have done this, how would the binding pose be changed? Would it be the same? Would it be slightly changed? Would it be completely different, or would we lose the interaction activity at all? It is very important to make accurate predictions on this step because the results from this step are an initial point for following simulations. For example, we use our tool based on molecular dynamics to evaluate complex stability and some other properties. So how can we determine which pose is correct? The only physically reasonable metric of it is change of Gibbs free energy. Gibbs free energy is denoted as G, and equals entropy minus temperature multiplied by enthalpy minus temperature multiplied by entropy. It's okay if you're not familiar with that stuff. We can basically treat it as just potential energy of a system. So regarding our problem, how can we use it? The free energy of two molecules that are not in a complex equals to G1 plus G2, which are separate energies of two proteins. When the complex is formed, the system changed itself, and its energy is changed as well. So now free energy is equals to G1 plus delta G plus G2, and this delta G is what we actually need. The lower delta G is, the more energetically beneficial complex we have. So for different poses, we have different values of delta G, and the lowest delta G corresponds to the correct answer, and that's what we looked for. Okay. Still, we have a lot of issues remained. The problem itself has very huge translation space because we need to evaluate a lot of molecules' positions that differs from each other just by position in three-dimensional space. The same situation is with rotation space, and in fact, we have deal with a Cartesian product of these spaces that makes the whole solution space even more huge. Delta G itself is computationally intensive, and it requires accurate structures. By accurate structures, I mean structures without gaps, without missing atoms, without stereo clashes, and so on. So there are additional requirements. And last but not the least, proteins are not rigid, they are dynamic, and some of them are more rigid, some of them are more dynamic, but in any case, we have to account how proteins are organized. Okay, the task is very hard, but nature and diseases don't care about huge solution spaces, accurate structures, and so on. Typical project of drug discovery requires to solve this task hundreds of times. So we must handle it somehow. Typical protein-protein docking pipeline looks this way. Let's first look at the last step. The majority of protein-protein docking tools use different scoring functions as a primary metric. Wait, but what about delta G? They don't use delta G due to issues that I talked above, due to its high computational cost and additional requirements to structures. But we do want to overcome these difficulties. So our docking pipeline looks very similar, except in the final steps. As a final step, we refine solutions with minimization algorithms that allows to make interaction surfaces of a molecule more complement to each other to avoid stereo clashes, and in fact it makes delta G metric usable in fact. So let's look at some details. To scan translation space very fast, we use FFT correlation theorem approach that came from images processing, if I'm not mistaken. In order to apply it, we need to transform our molecules into three-dimensional grids. In these grids, we account geometry of molecules, electrostatic forces, interactions with water. We do this for both molecules. Then we calculate correlation function of these grids using FFTs, and then take just best results based on this correlation function. Then we repeat this procedure for each rotation of the second molecule, and this way we scan rotational and translational spaces. QFFT library is used for FFTs. Then we take best by delta G. We use our own implementation of OPLS force field. It is deeply optimized for GPU. I spent a lot of time on optimizing it. Then we clusterize the results because there are a lot of very close results that are a bit different, and there is no need to account all of them. Then, as I said, we do energy minimization. We have carried out quite wide research on optimization techniques. We have evaluated about 20 different approaches, and Ribia-Pollock conjugate gradient method turned out to be the best. And then we finally take best results by delta G values. So fortunately, each step of this pipeline is really well parallelizable, and the whole pipeline work on GPUs. And moreover, we can process each rotation fully independently, and it allows us to parallelize it at a different scale. We implemented multi-GPU mode. We process each different rotations on different GPUs, and it allows us to scale linearly. We achieved significant performance speed up. On rigid docking, our tool works about 7 minutes on Tesla V100, while other state-of-the-art tools work about 90 minutes on 8 CPU cores. Rigid docking means docking without a minimization step. It has very limited application, but in some cases it could be used. On flexible docking, our tool works an hour and a half, which is equal to 90 minutes, in fact. The same calculation on 8 CPU cores would take tens of hours. To estimate accuracy, there are two well-known protein docking benchmark datasets. One is from Capri, which is worldwide protein docking competitions, and the second is protein docking benchmark. They are maintained by academical groups. I took 55 complexes from these datasets and got 80% accuracy, which is quite high and one of the highest in the field. Very nice. We developed a very cool tool. We can solve docking problem very well. We can optimize antibodies really well. It works, and it's nowadays reality. We do it on everyday basis. But what's next? Nobel Prize in Chemistry in 2017 was awarded for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. It means that year by year we can resolve more and more quality three-dimensional protein structures, and the same situation is with quantity of that structures. It grows exponentially. So we must use this data. Let's imagine that we have three-dimensional structure of a target, and the goal is to create medicine against it. We could design some groups of amino acids that definitely bind with the protein of interest. Then we can construct it further and further, and finally design the whole protein with desired properties from scratch. Very exciting, isn't it? If we were able to do this, we can drop some expensive and time-consuming stages of the pipeline and replace them by pure in silico steps. In order to do this, to reach this goal, we must develop more accurate and more fast bioinformatics tools, approaches, models, and pipelines. So, in conclusion, I want to highlight that for me, bioinformatics is the most exciting field. There are a lot of not solved problems, very big free space that could be filled in with very quality software, and working in bioinformatics we can contribute to the most serious humanity challenges like cancer. And so let's work on this, and we will defeat all diseases. Thank you very much.